Hi guys, welcome to the Bio93 lab tour / demo. I just parked my car and I'm getting ready to head up to the lab. So you may or may not recognize this building, but that is McGaw Hall where my lab is located. Here's a Starbucks coffee that we have right outside of my lab. Starbucks is my drug of choice. Alright, so in an effort to reduce my touch points, I pop out my key and then I use it to poke the elevator button because my lab is on the 4th floor. Same thing with the buttons inside the elevator. And my lab is conveniently located right here, just outside of the elevator. With COVID being a thing, we have to sign in every time we come into lab. Hey, there's Dr. Thompson-Pierre.
Alright, so to give you guys a general overlook of our laboratory, this is the space here. We have lots of equipment aligned along the walls in the back. This is a separate room that we use to keep some of the flies that we're currently quarantining. So when we get new flies, they sometimes have mites in the cultures. So I have a box of flies that I'm quarantining currently over there. Here is our lab microwave. So only lab things go in here, which I will show you later. And then this is our cafe area. So we have a Brita water filter, we have a Keurig coffee maker, we have our fridge, our microwave, a toaster. So we don't live in the lab, but we do spend a lot of time in here, so we like to be comfortable.
Here is my lab desk. I usually have my computer here, but this calendar generally stays here. And you can see I like to color code my calendar according to my experiments. And then I cross out the days as they go. Sometimes experiments get messed up and then I have to cross them out and cancel experiments, which sucks. So, yep. Here is a look at a page in my lab notebook. So something that you guys should notice is that I write the date first off that I'm doing these experiments. I write what the goal of my experiment that day is. And here's just a very simple pictorial representation of my animal. A means the anterior end, so it's head. B is the posterior end, so it's booty. And then I write down any notes that I have from my experiments that day, as well as any settings that I had on the microscope. So, yep. You can see I can flip through this. Those are all experiments. Sometimes animals die, so I have to cross them out. This is me trying to figure out genotype crossings, so lots of things can go into this lab notebook. Alright, so this here is my imaging kit, which contains a couple things that I'll need for my experiments. Number one, my forceps. This is the tool that I use to grab onto the larvae. And it's got a little cover here. Second thing here is glycerol. We place this on top of the larvae, so it kind of creates a suction with the larvae and the cover slip. If we don't use this, then the larvae just gets crushed. The second thing that I'll be using today is our cover slips. So this is what we place on top of the larva to kind of squish them into place, and they just squish into the agarose pad and they just chill. Here are the microscope slides that we'll be placing the agarose pads onto shortly. And I will also be using this, which is just like a glass thing that has little wells in it. I will place water into these, squirt some water in here, and then this is what I use to clean the larva. So moving away from my lab bench space, there's my desk. We will come over here. Over there is our fly room, and here is our dissecting scope that has a fluorescence laser attached to it. I'll show you guys that in a bit, but first let's look at these gas tanks over here. This is our carbon dioxide tank, which we use to knock out the flies. So we just use CO2. They kind of move around and freak out for a little bit, and then they're knocked out, so then we can move them around and sort them however we'd like. This is a nitrogen tank that our nitrogen and carbon dioxide tank. It's a mixed gas tank that another graduate student in our lab uses. She is trying to figure out how we can injure the dendrites of these animals using low oxygen conditions as opposed to the 2-photon laser.
Okay, so then moving into this room, this is going to be our fly room or our fly sorting room. This room has a whole bunch of different fly sorting stations. So this is one station here, we have another station over there, and then we also have one station right there, and a fourth station over there. This is the station that I like to use the most, so all of my flies are right here. These are flies that I have that are currently crossed. So let's take one of these out here to examine. So these are flies of one genotype, and these are female flies. I put how many female flies I placed into this vial. These are male flies of a different genotype, and I also wrote how many male flies I put into this vial. This is the date that I put them in. This is the date that I transferred them. And then this is the genotype that I want to pick out of the F1 generation, so the kids of these fruit flies. And I have some other indicators of what they should or should not have. So this curly O means that they should have curly wings, not straight wings. This TM6B means that they should have shoulder bristles, lots of shoulder bristles as opposed to just a little bit. And then all the flies should have red eyes. I transferred this vial today, so let's take a look at the flies that used to be in this vial. They are now in this vial. And the reason I transfer them is so that I can collect virgins from these vials eventually. So these are all the flies that I've collected already, and I can use them whenever I want to start new crosses. So here are all my males, here are all my virgin females. You may be wondering why I'm collecting virgin females. That sounds really weird, I'm sorry. But this is the symbol for female. This is, the little V on top of it is a symbol for virgin. The reason we collect virgin females is because Drosophila females can actually store sperm. So if they're storing sperm of a male that they had previously mated with, then when we cross them to males of the genotype that we want, then their offspring aren't going to be the genotype that we're expecting, because they might either use sperm from the new male or they could use sperm from the old male, which is kind of wild. This is kind of what I was talking about earlier. So here we have a fly with straight wings. Here we have one with curly wings. This is the shoulder mutation that I was telling you about. So we can see here there's not many shoulder hairs here, but then there's a whole bunch of shoulder hairs here. So that's that TM6 mutation. There's several eye mutations up there. That's a grumpy fella. That's you guys on exam day. That's you guys at the beginning of the quarter with broken wings. You don't know how to fly yet. That's you guys at the end of Bio93. You guys will know everything there is to know about biology. I'm just kidding, because I certainly do not know everything there is to know about biology, so it'll be a long time before you can do that. And before I can do that. I'm not even close. So those are our fly sorting stations. And then this over here is our incubator room. So incubators are essentially just refrigerators. It's a little loud in here. I'm sorry. I'm going to try to speak closer to the microphone. This is our 18-degree incubator. So you can see up here, it's set to 18 degrees, and it's currently at 18 degrees. So we have some fly stocks in here. The reason we put flies in here is because at 18 degrees Celsius, the flies grow very slowly. So their developmental time period is longer, which means it takes them longer to grow. We put flies in here whenever we want to keep them, but we're not necessarily like using them for experimental purposes. So since they're growing slower, we can wait a little bit longer in between times to flip them into a new vial, which is nice. This is another incubator that's set to 25 degrees Celsius. It's currently at 24.78, 0.7, and then it switched back to 0.8. So we place flies in here if we really want to do an experiment really fast. It's generally not good to speed up the developmental time window, because that could have different effects. But
And then here is our third incubator. This is 22.5 degrees Celsius. It's currently at 22. If you really want to control for the temperature that your flies are at, so room temperature might fluctuate like plus or minus 1 degree. If you really want to make sure that your flies are growing at the same exact temperature their entire life stage, you can place them in here. Alright, so walking out of the rooms with the incubators, the last thing I want to show you here is where we keep our unused fly food, which is in this refrigerator. So you can see there's no genotype on here. This is an unused vial. In there you can see some little white pellets. So those are just yeast pellets. And yeast actually encourages female Drosophila to lay eggs, because it encourages them and lets them know that there is a good food source available for their progeny to survive and thrive and do well, like you guys when you're done with Bio93. Alright, hello guys. The first thing that we do in terms of getting ready for this demonstration, where we're going to be cutting the dendrites of our larval neurons, is we have to make a little squishy pad for our animals to just chillax on while we're cutting their neurons. It sounds painful. I don't think it's very painful for them. However, I can't talk to larvae, so I don't know. So this is a vial of agarose. You can see that it's not melted right now because it's kind of like not very see-through, but that will change once I heat it up in the microwave here. So the way we do this is we just put it into a beaker of water. And when we heat this up in the microwave, it's going to heat the water, and then the water will heat up the agarose. So we'll put that in there for 1 minute and 15 seconds, and we'll check on it in a bit. This piece of equipment is very central to, I would think it'd be safe to say, probably every biomedical research lab. This is called a pipette. And pipettes, the function of pipettes is essentially to transfer small amounts of liquid in a very, very precise manner. So here you can adjust the volume that you're transferring when you're using a pipette. So the way this works is this is a P1000 pipette, meaning that it can transfer 1,000 microliters of liquid or of solution between beakers or vials or whatever. So when you have it to the max setting, which is here, the 1 is red, meaning that like you can't go above that. This is 1,000 microliters. When you have it set to 90, this is technically considered 900 microliters, not 90 microliters, it's 900. And so 80 will be 800 microliters, 70 will be 700 microliters, so on and so forth. I just keep it at 1,000 microliters because I don't really have to be precise with the amount that I'm transferring. So there's that. And then the part of the pipette that we use to actually transfer stuff is the pipette tip. So this is the pipette tip now, and they're stored in these little boxes like this. So now I have my pipette tip on, and lay that down. And then I can take my beaker out of the microwave. You can see that I've heated the agarose solution. So now it's pretty much see-through and it's just like a clear liquid. So what I'm going to do is I'm going to take some agarose from here, from my agarose vial, and I'm going to place it onto my microscope slide. And then I'm going to place one microscope slide with the agarose, or I'm going to place the microscope slide that does not have the agarose on top of the one that does have the agarose, so that I can make the little agarose pad for the flies. So in terms of using the pipette, what you do essentially is you press down, and then you dip the pipette into the solution, and then you slowly release up at the top of the pipette, and you'll see the solution going up into the tip. So that's what I'm going to do right now. So I'm collecting some agarose into my pipette tip here. You can see that I have some agarose in there. I'll put it onto the slide. Once I feel like I have enough, I just flip the other slide on top. I can toss out the pipette tip. This little button here pushes this thing down, and then it pushes the pipette tip out. And you can see I now have my agarose pad on my microscope slide. Perfect. Okay, so now that I have my agarose pad here, I can take my imaging kit over to the fly sorting stations, which is where we are going to be picking out our larvae and mounting them. Mounting meaning just putting them onto the agarose pad. So this is the fly sorting station that I generally like to use. So I will set up here, and you guys will be able to see me picking the larvae and transferring them onto the agarose pad that we just made. And then that's essentially just preparing them for us to do the injuries on them.
The first thing we do is we'll turn on the light so that we can look at our larvae. And these knobs here are basically to adjust the zoom and the focus. Here is our agarose jelly pad that we just made, so I'll lay that here. This we use to clean out our larvae, so there's like little wells in here, so I can squirt some water in there. Like I said, we'll use that to clean out our larvae. This we're going to be using to dry our larvae. So we can just fold this up like that. And when I'm using my forceps, I can place the larvae on here and it'll dry them off nice and easily. This is glycerol. We put a little drop of this on top of the larvae so that when it kind of creates like a seal in between the cover slip and the agarose pad, as well as the larvae. If we don't have this, when we place the cover slip on top of the larvae, it will just pop. So this is very important. here's our cover slips. So cover slips essentially look like this. They're very thin pieces of glass that we place on top of the microscope slide for imaging. And then I have my handy dandy forceps that I use to transfer the larvae, and I'm going to try to show you guys the larvae, but I'm not sure if you'll be able to see them with the naked eye. Our larvae that I collected earlier for you guys is in here, and again I collected that doing the egg lay with the flies. And yeah, that is all we need to cut. So I'm going to go ahead and demonstrate how we do that. So first, we place these guys under the microscope here, and then I'm going to look through my eyepieces, and I'm going to adjust the magnification and the focus so that I can tell what I'm looking at. So I can see the 3 larvae that I placed in here earlier, and I'm just going to grab one of them out. These are really, really tiny, so I'm going to zoom in quite a bit. Okay, you definitely won't be able to see that, but if you see a little white tip on my forcep, that's the larvae, very tiny. And I will place it in the water. They don't drown, they just kind of like wiggle around, so he's just chilling. He's in Palm Springs on a hot day or something, I don't know. So I'll whirl it around, because right now it was like crawling around in the yeast, so it's full of yeast inside. Another handy dandy trick that I do to get rid of all the yeast on the larvae is I'll pull it out of the water and then drop it back in, and the sheer force of it like being placed into the water kind of removes all of the yeast from around it. Think of like you being super sweaty on a hot summer day and then you jump into the water and all the sweat comes off, kind of like that. Okay, so once I'm pretty confident that all the yeast came off, and you can see me playing with this focus knob right here, it's because the larvae is constantly moving up and out of the water as I'm trying to clean it. So once you're confident all the yeast is off, you can pull it off. So now you can see 2 little water droplets in there, or in between my forceps, and the larvae is in one of them. It looks like it is in the right one. Again, you can't see it, but it's in the right drop, in the right droplets on this side. But I'm going to go ahead and dry it off. And it was actually in the left droplet, which is why it's important that we work under this microscope to see the larvae, because they are very, very tiny and they're very, very hard to see. Okay, so now I have him here. Now I can say with confidence he's on the right tip of my forcep. I can see him with my naked eye, so hopefully you guys can too. If not, it's okay. So now that he's cleaned off and dried off, I can go ahead and expose my agarose pad. I don't want to expose it too early or else it's going to dry out. So now that I'm ready to place the larva on here, I can go ahead and take off that top slide. And I will just place the larvae on top of the agarose pad. And it is just chilling on there. I try to put it as much in the middle as I can. If I put it too much on the side, when I place the drop of glycerol on it, it could fall off the agarose pad, and that's not good. It's just a headache. So I will place a drop of glycerol just on top of it. And then here I'm going to use my cover slip to just place this on top of the larvae. And the larvae will essentially just smush into the agarose pad, and it's just going to be chilling. Probably a little bit chilly too. It's cold these days in Irvine. So right now it's smushed in there, but it's currently kind of like on its back. So I'm going to rotate it so that its back faces us, and we can see its neurons super, super clearly. So I'm pushing, 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 pushing. And now I feel like it's in a good place for me to show you guys how to cut the neurons. Okay, so I still don't know if you can see it, but the larvae is on like the left side here. Again, it's important that you place it in the center for this reason too. When you put the cover slip on, sometimes it like moves over to the side, and then it can fall off of your agarose pad, which is never fun. So yeah, that's how you mount your larvae. Our larvae is now mounted, or our larva, I should say, larvae is plural. Our larva is now mounted, and we are ready to take it to the microscope, to the 2-photon microscope, to use the 2-photon laser to cut its dendrites. Most exciting part. Alright guys, so this is what we call an inverted microscope. I think this lighting might be a little too harsh on my face, but you can see that the objective lenses are located at the bottom, and then this part up here is basically just a camera that feeds into the computer, so we can see our neurons on the computer, and we'll get to that in a little bit. So since this is an inverted microscope, what we're going to have to do is, if our sample is on the top side here of the slide, and there's nothing here on the bottom, instead of putting our slide on it like this, we're going to have to flip our stuff upside down, so you can see that the slide is there on the bottom, and we're going to have to put our microscope slide down like this. And let me just confirm which objective. We're using the right objective, so we're using a 40x objective, and it's an oil objective, meaning that I have to put some oil onto the specimen. The specimen being my little fruit fly larva. So I think you might be able to see it, but it's this little white dot that's like right here on that side. So I'm just going to put some oil on it right now. It's like an air bubble. Another air bubble. Finally, a good nice drop of oil. So we can see the oil on there. That big white thing that you see is another air bubble, but our larva is somewhere in that drop of oil. So now I'm going to go ahead and place this down onto the objective that you see there with the pink little band around it. And again, I'm placing this upside down because this is what's known as an inverted microscope. So the objective and the light comes from the bottom up, and we want it to be shining on our specimen, not our microscope slide. So I'm going to lower the stage all the way, and then I'll place our microscope slide on there. And then I can essentially use this little joystick to move the stage around. So I'm going to move it so that it is just over my larva. Okay, so I'm pretty confident that it's just over my larva there, and then I'm going to raise the objective lens up until it makes contact with the oil. I'll move this down. Right now I'm going to try and find my specimen. So I'll let you guys see the computer screen a little bit. I think I can capture this angle pretty well. Let me keep it here. So right now I'm going to try to find my larva. You'll be able to see on the computer screen because remember, the top part here is a camera. The bottom part is where the lasers and the light shines so that we can see our animal. So first I'm just going to do a bright field light, which is just regular light microscopy. So I'm just trying to find my animal first and make sure that I'm in the right Z-stack. Okay, so it looks like I found my animal. So essentially I just move the objective up and into the animal. And I can show you what that looks like over here. So let me make sure this animal has neurons first. So we have to make sure that not only we can find it, but that it also is the right genotype to where we can see its neurons. And yep, I can see its neurons under here, which means that we are ready to look at some animals. Alright, so from now on you guys are going to look at everything that I'm looking at on the computer screen. So we found our animal using the locate function for the microscope, and now we're going to look at it using the camera feature. So here is live imaging of the animal, and we can already see some dendrites there. However, this is like super zoomed in, so I'm just going to reset the zoom them all the way out. So here we can see our animal. This is the larva that I mounted, or that we mounted earlier. So here is the head of the larva, and you can see all of its neurons in there. And then as we move down, you can see the tail of the larva, and all of the neurons throughout the larva's body. Not all of them, but the ones that we're interested in. So I'm going to move back up. Okay, so we're going to pause the live imaging here. This here is an example of a neuron I cut a little bit before. So here you can see the neuron before I cut it, and then here you can see the neuron after I cut it. So again, this is the cell body of the neuron. I think you guys can see my pointer. This isn't zoomed, so obviously you can see my pointer. So here's the cell body of the neuron. This darker area here in the center doesn't stain. So this darker area here is actually the nucleus. It does stain, but not with the genetic construct that we're using. So here's the nucleus. You can see the dendrites all around the cell. And then this little piece here that goes down but then eventually disappears is the axon, because the axon actually goes into the animal and eventually disappears. So it's not in the same Z-plane as all the dendrites are. So think about that as like if the plane of the neuron were like my face, right? This is like, my face is like relatively flat. But my brain is in a different Z-plane. So my face is in this Z-plane. My brain like probably starts around here. So this is like where the axon goes down into, and the axon will eventually go into the animal like that. So that's what I mean by different Z-planes. Once we injure the animal's dendrites with the 2-photon laser, it kind of creates these like craters that you see here on the right. So I cut the dendrite here, I cut it here, looks like I cut it here, here, and here for a total of 5 cuts. You don't want to cut it too close to the cell body because then the neuron will just die. So you have to be careful with that. And then you also, when you're cutting dendrites that are close to the axon, you don't want to cut the axon because that will also be bad for the cell. So you see I have a second container here, and this is where we're going to have proof of our injury, or evidence of our injury. And then I have this container here, which I'm going to be using to image. So we're going to go into, instead of live mode, we're going to go into what's called a continuous scan mode, which just gives us like a better image. So we will go into continuous scan here, and I am just going to choose any neuron, pick a neuron, any neuron. Let me find one that I like. I got my eyes on one of them. I'm just trying to move. I'm trying to find one that's nice and flat, so that we can see the whole dendrite arbor and the cell body together. Don't know if I'll have any luck with that, but let's see. Okay, this one looks pretty nice. So I've identified the neuron that I want to cut. I'm going to crop it, or crop the imaging window so that it's just on my neuron of interest. And then I'll do continuous again. So now we can see our neuron here. We can see all the dendrites of our neuron. Here I can move through the Z-plane, so I'll move a little bit deeper into the animal, and there you can see the neuron. I'll move even deeper into the animal, and you'll start being able to see the axon. So here you can see the cell body, and then here you can see the axon. If I go deeper, oh, it looks like the axon is like right down here. Disappeared again. Let's see if I can find it. Yep. So the cell body is somewhere over here, and you can see the continuation of the axon down here. And the axon of these neurons go to what's called the ventral nerve cord, which is kind of like our spinal cord, but for the larvae. So let's go back to the Z-stack where our cell is. Okay, so there's our neuron. And then we'll go back to where all of its dendrites are. Ta-da! So there are all of the dendrites, and there's the dendrite tree. So now what I'm going to do is I'm going to focus this imaging window to a very specific position that I want to cut. So let's say I want to cut this dendrite branch. I'm going to focus that window super tiny right there. I'm going to click on this side, and then I'm going to adjust the laser power so that it's super strong, and I can essentially blast the dendrite and kill it. So here I'll set the laser power from 17 to 30. I'm going to slow down the laser speed, change the imaging resolution, and then I'll hit continuous, and you're going to see it start going on this side. And when we see a bright green flash like that, that means that we've successfully injured the neuron's dendrite. We can go back to the first window on this side. We can hit reuse, and this will reset the laser power to just like the regular imaging percentage. And then we can take a picture of it, and boom. So we see that we created that crater again right here. We can actually zoom this in a little bit more. So we'll go to 3 zoom. So we can see that we created that crater that I showed you guys from that image before. So remember, this was the after picture, after injury, and you saw these craters and this was a neuron that I did a while ago. So this is a neuron that we just did in real time. We saw evidence of the injury, and then we got that crater over here, just like we did in that other picture. And then, oops, I did not mean to do that. That's okay. So then what I'm going to do is I'm going to get an updated snapshot of this neuron's dendrites, and we are going to find a new location to cut. So this branch here looks nice and juicy. We want to cut the branches wherever they're showing maximum fluorescence, because that means we're in the right Z-plane. So I'll focus in right here. Again, I'll crank the laser power up, move the laser speed down, change some of the frame size here, click on the right side, and then I'll continuous again and wait for that bright green flash. So we see it here, we can stop imaging, go back to our first window, reuse the settings from this picture, and we can take another picture to confirm the second injury, which we see right here. So now we have the first injury right here, and then we have the second injury right here. Okay, and then it looks like there's one more place that I can cut this neuron's dendrites at, and it's at that bottom left location, so I'm going to try to find a nice juicy Z-stack for that. And it looks like this is the winner. So I can cut it right here. Crop that, again, crank the laser power up, laser speed down, change the frame size, and then wait for that bright green flash of fluorescence. This one's taking longer. There we go, now we see it. And then, oops, that is not what I meant to do. Let's go back on this side. So what I did there was I told the system to snap a picture without hitting reuse first. So that messed up all my settings, but it's fine. I can just manually reset them. My neuron might be slightly out of whack. Just a little bit out of place. So we will move it. Yeah. So that's nice and centered again. Okay. So we cut at 3 different points, and something that I want you guys to notice is that we can see what's called beating of the dendrites, and this is something that's very characteristic of dendrites that have been recently injured. So we can see the dendrite beating occurring here. So we see all these little like essentially they're like little beads or circles in the dendrites, which means that the dendrite is actually dying and degenerating. Again, we can see evidence of injury number 1, injury number 2, injury number 3. I wish I had taken a before picture. I didn't do that, and I should have. But we can compare it to a neuron before injury, right? So this is one before injury. We see no beating. See, we can see the dendrites nice and clear. And again, this is one after injury. We can see all this beating and the dendrites, meaning that we've successfully cut this neuron's dendrites. Yeah, so then what we do is in the subsequent days, we assess the neuron's regenerative potential. So we just take it to that other microscope that doesn't have the 2-photon laser, and we essentially just look at how much the dendrites have regenerated. So I'll check it tomorrow to make sure that I made all of the cuts that I needed to make, and then I'll check it in 72 hours from today, so in 3 days, which would be Monday, Tuesday, which would be Wednesday, to look at how much the dendrites have regenerated over a specific amount of time. Anything else? Oh, I can show you guys also how we kill a neuron. So this one we cut, so we're going to reset our parameters, our like zoom parameters. Zoom, so this is the dendrite this is the neuron whose dendrites we just injured. This is the one that I had zoomed into. Now I'm going to move down and kill this neuron that's down here. So let's center out on this guy. Okay, so for this, for killing the neurons, we want to go to the Z-stack where the neuron's cell body is. We don't want to go to where the dendrites are, so I can move deeper into this animal to find the cell body, which you see right there. For killing the cell body, it's essentially the same thing as what we do to cut the dendrites. We just focus this, the little crosshair onto the cell body. And again, this is the axon coming down here, and then you can see it a little bit again right down here. So focus the crosshair onto the cell body, click onto the other side, crank the laser power up, and then we'll see evidence of injury on this side. Yep, so that bright flash that goes on like that, that's evidence of our injury. Go back to the first window, and then we'll hit reuse and snap a picture. And boom, you can see right there, that's like a little crater, and you can see the nucleus got a little bit deformed as well. So that means that we have successfully killed this neuron. If we look at it in a couple of minutes, its dendrites will actually show that blubbing effect, or that dendrite beating effect. You can kind of see it starting to happen here. It looks like there's some beating here, it looks like there's some beating right here. You can kind of see it here, and if I let this run, it's just going to the beating will get worse and worse. So we can see that when we kill a neuron, one of the first things that happens is that the dendrites start beating. So the neuron is sending some kind of signal out to its dendrites and telling its dendrites like, hey, you're no longer useful, I can't do my job anymore, it's time to kick the bucket and end our life here. Which is very sad, but it is what it is. So yeah, again, we can see some beating. Let's see if I can get a better Z-stack. Just like I mentioned earlier, some beating here, up in here. And yeah, that is what we do for our experiments. Okay, so then when we are all finished, we can just raise this. We can take our specimen out. And we're going to clean the oil objective that we use with some lens cleaning paper. So this is a very special paper that's designed to like not scratch the lens at all. So I fold it one time, then 2 times, and I just kind of gently stroke the objective lens like this to get out all the oil. Not sure if you can see that darker spot, but that's the oil that we had placed on the, yeah, you can see it, but that's the oil that we had placed on the cover slip. Alright, and now that we are done cutting the larva's neuron, we can put it back. Normally what we do is we place it into its own grate plate so that we can keep track of which larva was which. But I'm just going to put it back with its friends so it can chill out. It's had quite a stressful time being cut. It's a slower process because I'm trying to show you guys. So essentially I'm going to use my forceps to grab onto the cover slip and lift it gently so that I don't pop the larva. If I lift it too fast, the larva will just pop and die. sad for the larva. So I think, okay, it's still down. Yep, and it's alive, yay! It's alive for now. Sometimes the larvae are alive, and they're swimming around in the glycerol when you're done with them, and then you look at them either the next day or at the 72-hour time point, and they're just spontaneously dead, which is really freaking frustrating. And sometimes the ones that look the weakest actually end up surviving, so it's kind of a crapshoot when it comes to that. So I will lay this guy back down into the yeast, and he is happily swimming along and living his best larval life. Okay, so for this next segment, I'm going to briefly show you guys how we collect flies that we want to collect for certain crosses. So remember I showed you that I have all of my males and virgin females up there in those 3 boxes, and then I have all my flies for my crosses in this box. So how do I collect all of these flies so that I can get them ready for the crosses which are over here? So that's what I'm about to show you guys. Alright, so step 1 is turning on the CO2. I already have it on, but essentially I turn this valve to open it, and then this is in the closed position, and I open it like that. And then when we go over here, okay, so remember this is called the fly sorting station. When we're at the fly sorting station, we can turn on the local CO2, so you're going to see a bubble pop up over here. So you can see that our CO2 is on, and it's running through both our needle here that we insert into the fly vials to knock them out, and our sorting pad. So there's CO2 that's constantly being pumped up out of this pad. So when I place the flies on here, they're going to remain knocked out while I label new vials to put them into. So once again, say hello to my children. This one's Tony, this one's Sam, this one's Cindy. No, I'm just kidding. Let me see if I have any that have recently eclosed. If not, I'll just do like a random demo. So eclosion is what it's called when they come out of these little, we call them pupae, or they're like shells. So in here you can see some flies. There we go. You can see some flies that have not eclosed yet, which are like the light pupae, and then you can see some flies that are about to eclose, which are these darker pupae. And then you can actually see some flies that have eclosed, so you can see this one here since this morning. So the way this process works is we clear the vials in the morning, and then we collect them in the afternoon, because the flies don't mate for the first 8 hours of their life. So I know that any females that I collect from this vial are going to be virgin females. And remember, I said that's important because the females can actually store the male sperm, aka their genetic information. And so when we're going to set up the females that have a certain genotype with the males that have a certain genotype, we want to make sure that the females aren't using sperm from like old males that they had intercourse with previously. Intercourse, yes, flies have intercourse. Yeah, I won't say anything else on that. Okay, so the way we knock them out is I insert the needle into this little piece of cotton here, so you can see that the needle is in there, I think. Yep. Okay, and then I'm going to open this valve so that it knocks them out, and there's a fly there on the white. You're going to see it fall eventually. Yep, I think you saw it fall. I'm pretty sure. I'll check the video later. If not, they all fell to the bottom. So now I will take this, and I'm going to dump it onto my fly sorting station. Alright, so there's one little sucker, there's 2 little suckers. So I've only had 2 flies eclose. I actually checked these earlier today, so that's why there aren't many. And I don't think I have any others to collect virgin females from yet. So we will just collect these 2 if they are of the right genotype. So what I'm looking for here is that the flies have curly wings and that they have extra little hairs on their shoulders. So this is a male who does fit that criteria. And then this is another male who fits that criteria, which is great because that means I can collect them. So I take a fresh vial with fly food in it, and I'll scoop these guys into there. They're currently knocked out. You can see them at the bottom of the vial there, not moving much. And I'm going to label this vial with their genotype, the date, and then whether it's a male or a female, so I don't have to like look closely at them. Okay, so this is, again, what typical labeling looks like. So this is the genotype of the flies here along the top, and then this is the date that I collected it, and then this is whether it's a male or a female. So this is the symbol for male, the symbol for female. I've shown you before, so I won't show you again. Those are the only flies that I can collect right now. And you can see they're already starting to wake up. This one in the back is starting to crawl up the wall of the food. The one that's in the middle still is just chilling. Let me try to wake him up. Oh, there he goes. He's up. I startled him. So now he's up and back to moving. So yeah, they wake right back up after, like, I don't know, a minute or so of not being on the CO2. So I'm going to place him up there with all of the other males up there that I have collected. And that is essentially how we collect flies here. Okay, so that concludes the lab tour / demo of me cutting our Drosophila neurons. I hope you guys enjoyed that. I hope it was informative. I hope you get the question that comes from this demonstration right on the exam. It should be a free point because I don't think the professors are going to try to knock you off too hard. I guess some points to remember is that we used a 2-photon laser to injure the larva's neurons. We injured their dendrites. I'm trying to think of what else. Oh, they can ask a question about it being an inverted microscope, meaning that the light is coming from the bottom up and the objective lenses are on the bottom, the camera is on the top of the microscope. I'm not sure what else they could ask you about. They could ask you about the pipette and how that works, or the function of the pipette, which is, remember, to transfer very small amounts of liquid from one container to another, because you can imagine that'd be really hard to do with, like, a beaker if you're just pouring a beaker into another beaker. So we love pipettes in biomedical science laboratories. They can also ask you what each of the temperatures in the incubators do, such as the 18 degree Celsius incubator versus the 25 versus the 22.5, and anything else that was covered in the video, I guess, is fair game. I didn't go too much into depth about what we do. I don't think they're going to ask you, like, a super nasty question that's going to ask to build on extra knowledge, but yeah, hope you guys enjoyed. If you guys have any questions, like I said, I'll be one of the panelists in the room live on Friday, so you guys can ask questions in the Q&A or in the chat, and I'll be happy to answer them. And yeah, I hope that was informative. I hope this kind of gave you a little sneak peek of what being in a lab is like, since you guys can't physically do it in these times. And hang in there, stay strong, get through college, don't leave bio. Bio is super fun, super interesting. I hope that I, as a TA, at least for my discussion sections, have positively impacted a lot of your guys' perspectives on biology and have motivated you guys to pursue a career in biology, whether that be medical school, grad school, master's, PhD, going into industry, whatever you want to do. And I hope I generated some excitement because biology is so freaking cool. And that's all I have to say. All right, bye guys.